Suppose. Well, good evening, everyone. On behalf of the Department of Chemical Engineering at the Institute, let me welcome you to the 12th Distinguished Institute Lecture in the memory of Professor C.V. Seshadri. We're delighted to have Professor Narendra Dixit from IISc here. And before I formally introduce our speaker for today's talk, let me first request Professor Mahesh, our head of department, to welcome our speaker and also say a few words about Professor C.V. Seshadri, on whose memory this lecture is instituted. Here you go.
Very warm welcome to Professor Narendra Dixit. In fact, he's been here since yesterday. He gave a talk in chemical engineering department. Surely we look forward to his talk today on immunology. So before we start, a note about the late Professor C.V. Seshadri. So the late Professor C.V. Seshadri was a distinguished chemical engineer of the country. He did his PhD from CMU, that is Carnegie Mellon University, USA, followed by a research associateship at MIT. He joined IIT Kanpur as a young assistant professor in 1965, and he is known for his book that he wrote with S.V. Patankar on elements of fluid mechanics, Prentice Hall of India, 1971. In fact, I was looking through that paper. It's available for free for download because I think it's out of print, but it's a very interesting fluid mechanics book, especially for the undergraduate students. CVS left IIT Kanpur in 1974 to join Kasturi Paper Food and Chemicals Limited in Bangalore, where he set up India's first fodder yeast plant. In 76, he joined the Sri A.M.M. Murugappa Chettiar Research Center in Madras as the founding director, an institute emphasizing appropriate technology, a forte of CVS. It was here that CVS really blossomed and helped develop several appropriate technologies, including that for Spirulina algae. CVS also developed a catamaran for fishing made of HDPE pipes for the fishermen there, which was sealed on both ends, which did not rot nor attract termites. Thus, CVS was talking about and living innovation as early as the 1970s, which is a catchword in the present times. He lived quite a simple life near the seashore in a cottage like in villages, having no doors. For his efforts in this direction, CVS received the prestigious Jamnalal Bajaj Award for Science and Technology for Rural Development in 1981. Rajni Bakshi has written a nice biography on CVS. I think it's available online. And in there, she sums up his life's work. She says, CVS's youthful zest and enormous energy made it easy to forget the linear dimension of his mortal frame. Yet this is all the sea snatched away. I'm told that he went out in the sea as he would do every evening and unfortunately did not come back. So the man's bequest remains, awaiting the nurturing care of fellow travelers in this and other times. CVS received enormous support and encouragement for his efforts from Mr. M.V. Murugappan, with whose vision the research center was set up. I would now request Professor Supreet Saini to introduce our speaker, Professor Dixit.
Thanks, Vish. Again, it's a pleasure to introduce our speaker and our friend, Professor Narendra Dixit. He is a faculty in Chemical Engineering and Bioengineering departments at Indian Institute of Science, Bengaluru. And he is a graduate of our department after completing BTech from IIT Bombay in Chemie. He did his master's and PhD at University of Illinois Urbana-Champaign, and after a postdoctoral stint at Los Alamos, he started his group at IISc in the year 2005. And the focus of his group has been working on open problems in sort of modeling infectious diseases, particularly viral infections and their interplay with immune systems, and more recently some of the work on modeling and understanding how microbiomes behave. His work has been published in journals such as PNAS, Cell Reports, and he's been recognized by agencies such as Indian Academy of Sciences, Indian National Science Academy, and the Wellcome Trust TBT India Alliance. His group has made significant contributions in enhancing our understanding of the interplay between viral dynamics and immune system dynamics. And in addition to the talk that he gave yesterday on the other pillar, his today's lecture is focused on some of the general principles and learnings from that branch of the work that his group has done in the last 2 decades. Thank you so much for accepting our offer, and we look forward to your lecture.
Good afternoon, everybody. Thank you very much, Supreet, for those very kind words of introduction. My thanks also to the department, particularly Professor Mahesh, and all of the people involved in inviting me here. It's been a great pleasure. I've loved the last 2 days interacting with all my faculty colleagues, who are also friends, like Supreet has said. And it's also great to be back to the department where I spent 4 lovely years. Much remains the same. I think you can sense the energy the moment you walk into the doors. So it's been great coming back. I never had the opportunity to meet Professor C.V. Seshadri, but I did read the biography that you mentioned, and it hits you. One can't but be called upon to marvel at the man, because he, in my opinion, represented so many different dimensions that we as faculty members aspire to imbibe in our own personalities, that you recognize that the man was made of sterner stuff. Right? So he was, like he said, he was somebody who wrote books on basic fluid mechanics. He also had a take on thermodynamics, which challenged the paradigms that we hold today as sacred in terms of the laws. And he argued, correctly or incorrectly debatable, that these laws had their origins in colonial value systems. And then he went on to do other things, start institutions, build companies, make products which are even today sold. And more than anything else, he had this tremendous feeling of, or desire, if you will, to uplift the masses here, including trying to free them from what we would today call the colonial mindset, many decades before all of this could become mainstream. So it's interesting to read what the person did, and perhaps it was unfortunate that we lost him in this tragic sort of swimming accident. So it's great for me also to be here to commemorate Professor C.V. Seshadri. It's wonderful that IIT Bombay is keeping his memory alive, and today I have the opportunity of participating in this celebration of his life's journey. I thought today I would talk a little bit about the design principles in immunology. Like what Supreet said, these are things that occur to us as we look at how the immune system functions, especially in situations where it is not very successful in dealing with some infections. For example, HIV, something that you cannot get rid of by yourself. In fact, there are no vaccines today despite many decades of research. Right? So there are some challenges that we understand still pervade our immune system, and yet we also know that for the most part it works beautifully. So what is it that allows it to work well in many cases but fail in others, and where it fails, what are the causes that trigger those failures, and are there ways in which one can address them? So these are the sort of thoughts that I'm going to take us through in the next 40-45 minutes. Just to put ourselves on the same boat here, the first thing we recognize is that this is a rather complex system. And you will see that there are therefore principles that emerge that are appealing to engineers, because there are ways in which parts integrate, they work in tandem, there are control mechanisms that allow things to happen optimally, and so on. And so these are interesting observations that one can make from the way the immune system works, especially given that there are so many different parts. So the immune system, first of all, pervades the body. There are many important organs like the thymus, the lymph nodes, the bone marrow, and so on. And these get activated when we are challenged by pathogens. That side, is it? You prefer me to use the other side? Brighter, okay. So we'll try this. And then you mount a response. And very broadly, this response has been divided into an innate response and an adaptive response. And already you begin to see signs of these designs that I was talking about. This innate system is a broad, quick, and dirty response. It doesn't recognize pathogens specifically, but it recognizes features that are common to many, many pathogens and mounts a quick response to prevent massive damage that could otherwise occur. But then an adaptive response is called upon, which is specific and targeted to the pathogens that one is faced with, and then it mounts a carefully orchestrated, controlled response which both kills the cells perhaps that might get infected by these invading pathogens, which happens through these things called the CD8 T cell responses, and also produces antibodies which don't allow cell-free pathogens to cause infections as much by coating them with these antibodies and asking our immune system to filter them out. And eventually when all of that is done, you are left with memory cells which then have information of this attack that one was faced with, which can then be used to recall a response that was previously mounted successfully and do that more aggressively and effectively much before what would have happened otherwise. So this is an interesting already design of why there is this partitioning, for example, and how it's optimal and how this memory is retained and how that can be harnessed through things like vaccines. So what I thought I'd do, and there are many, many design principles here, so I won't have the opportunity to talk about all of them, but I thought I'll touch upon a few that have emerged in our own work in understanding some of these aspects. So what I thought I'd do is touch upon 4 of these so-called design principles that emerge, and I'll talk about the different aspects of the immune system also in the process. I'll first look at a problem of this innate immune response and there being a double negative feedback loop. And I will look at this in the context of hepatitis C virus infections. Then I look at the CD8 T cell response and how it is involved in this dynamical motif that we uncovered that governs the outcomes of viral infections. And I will do this in the context of what are called lymphocytic choriomeningitis viruses and other infections. Then the other arm of the adaptive immune response, which is the antibody response, and interestingly this is the arm that learns our pathogen, and its response improves with time, becomes more and more specific to the pathogen that we encounter. And I'll talk about this in the context of HIV infection. And lastly, I'll see how we can touch upon memory, looking at what we call the immune repertoire and vaccination in the context of COVID-19. Okay. So let me start then with this innate immune response. Hepatitis C virus infection is today not so serious a problem, but not very many years ago, it was a very serious problem with many hundreds of millions of people infected. And it's not so serious a problem today because there are very powerful drugs. But these drugs came into sort of effect, or became widely approved only about a decade ago. So before that, this was a very, very serious problem. And what happens when you catch this infection is the following. About 80% of the people become chronically infected. So you can't get rid of the infection by yourself. 20% do get rid of it spontaneously. And then if you don't treat the infection, you develop very bad liver conditions called cirrhosis, and then eventually some percentage develop liver cancers. So this is a normal liver. This is just to tell you how bad the disease is. This is what a liver with carcinoma looks like. So this has to therefore be treated. But the question now is, what does the innate immune system do? How do these 20% people get rid of it spontaneously? What happens with the remaining 80% people? And how something can be done to improve these numbers? So the innate immune response in the case of hepatitis C involves a molecule called interferon. Right? Very simple process. This is the virus. This is a cell, let's say a cartoon of a hepatocyte, a liver cell, which is likely to get infected by this virus. Following infection, this virus uncoats its genomic content, which is in this RNA form. And in the cell, in the cytoplasm, there are enzymes that convert or make copies of this RNA, and it produces in the intermediate process, this copying process, a double-stranded RNA. Now this double-stranded RNA is a motif that doesn't exist in our body by itself. Right? So it's something that is foreign to us. So our immune system has learned to recognize this. These are called pathogen-associated molecular patterns. So you mount now a response. You don't know where this is coming from, but you can sense that there is a double-stranded RNA. And that's enough of a trigger for us to mount an innate immune response. So this is sensed by sentinel molecules. They trigger the production of interferon. Now this interferon is secreted out, right? And once it's secreted out, it also, it's a signaling molecule. It binds what are called receptors on the same cell surface and also surfaces of neighboring cells. When this binding happens, it triggers a massive signaling cascade called the JAK-STAT pathway. Now the downstream effect of this is the expression of several hundred genes called interferon stimulated genes. So it's a massive carpet bombing response. These genes block the replication of this viral viral genomic content. So this halts this replication process and protects this cell, converts this cell into some kind of an arrested state so that no further replication is possible. This is the way these interferon responses work, right? But this virus continues to thrive. It doesn't get rid, so this response doesn't get rid of this infection in 80% of the cases. So what really happens? In many cases, this sensing is poor. Sometimes the production of interferon is not adequate, right? And therefore you continue to have this replication going on. And therefore you use interferon as treatment. You add interferon as part of drugs and see if you can then trigger the same cascade. So this is therefore the treatment that was standard until very recently. So you now pass interferon as treatment, you trigger the same cascade, right? And you now prevent this replication and hope that there is cure. This works in some people but not in a whole lot of others. So if you now look at the responses to interferon treatment, this is what you find. What is shown here is the concentration of the virus in the blood of individuals as a function of the time following the start of treatment. The responses are highly variable, right? You see some people getting rid of the infection very quickly and they attain a sustained virological response which lasts many weeks after the treatment is stopped. In others, you see this control, but then there is a relapse after you stop treatment. In yet others, there's only a partial response, and in yet others, there's hardly any response, so the treatment is not really working at all. And overall, it's only about 40% of the individuals who are treated with this interferon-based treatment who respond to this treatment fully, get cured. So the question now is, what happens to the remaining? Why does this thing, if despite triggering the innate immune response, not work so well? So we looked at this a little more carefully, and it turns out that there is this double negative feedback loop that you can identify. So there's a little bit of biology now, if you can bear with me. This is the JAK-STAT pathway, and there are these genes that are getting expressed, hundreds of them. One of those genes is called protein kinase R, I'll call it PKR. Every time all of these genes are getting expressed, there is this hydrolyzation of this other protein called EIF2 alpha. This is an eukaryotic initiation factor, just another protein. Every time this translation happens, you have this GTP part hydrolyzed to GDP. But for this translation to be sustained, you need this GDP to be recycled. So there is another enzyme, EIF2B, that recycles this. So now this translation can go on. In the presence of this PKR and the presence of this double-stranded RNA, you have this PKR getting dimerized. So this dimerization is catalyzed by the virus, and this dimerized PKR is autophosphorylated. Now this phosphorylated PKR, it turns out, can phosphorylate this EIF2 alpha GDP. When that is phosphorylated, it ceases to be a substrate of this particular enzyme. So this recycling gets halted. And secondly, this phosphorylated entity binds to this enzyme and sequesters it. So now whatever little EIF2 alpha GDP is left cannot be recycled because this enzyme is also gone. Because of this, this translation of these ISGs is now blocked. So this is how the virus hijacks the interferon system. Right? So now if you think about this as an overall process, you will see that there is this double negative feedback I was talking about. These interferon stimulated genes would like to suppress this replication process, but through the PKR mechanism, these double-stranded RNAs will prevent in turn the production of these ISG proteins. When you have this double negative feedback loop, and we as engineers know how to analyze these systems, you will find that there are two different steady states that these systems can exist in. If I think about this as A and B, A high, B low, B high, A low are the two states that you can exist in. And you can show through detailed modeling of all of the kinetics that you actually get these two steady states. There are these two Q1 and Q3 are stable steady states, and in between there is an unstable steady state. What are these steady states? In one case, you have very low interferon stimulated genes and very high copies of the virus. So this is where the virus has won over the interferon system. And in the other case, you have very high levels of these ISG proteins and very low levels of the virus. So here the cell has been protected by the interferon system. Now which of these steady states you go to depends on your initial conditions. If you're on this side of this intermediate boundary, you'll probably go to this state. If you're on the other side, you'd go to this state. So now you can ask what happens when you have a whole lot of cells, hepatocytes in the liver, which are subjected to this double negative feedback loop. And you can see, so by the way, before I do that, you can already see why this paracrine signaling, where interferon stimulated by one cell helps protect neighboring cells, because there you have now interferon genes upregulated before the virus has entered in. So you're moving the initial conditions towards this state. So you're more likely therefore to be protected in the neighborhood. Anyway, so now you have a milieu of cells that have different levels of expressions of various proteins, different levels of efficacies of interferon, different levels to which these double-stranded RNAs can block interferon production and so on. So if I lump all of that, and which can be done also formally, into a net interferon effectiveness, then I can see that from this double negative feedback loop, I get three different types of cells. There's one set of cells which have interferon effectiveness very low. They only allow this virus high steady states. You can have very high interferon effectiveness in some cells. They only allow these clearance steady states, and in between you have cell types which allow both these steady states, and the steady states that you realize would depend on your initial conditions. Now, depending on how many of these cells are there, your response becomes different. These cells depending on how many of these cells you have, these cells get cured. These cells get protected after some time, because even if you have the virus in them, they get killed, and so you don't worry so much about them. But if you have a lot of these cells, then you can't respond very well. So one can construct a model of virus dynamics, and you can show that depending on how many of those cells you have of this type that admits only the Q1 state, you can recapitulate all of the different dynamics of the treatment response that we saw. So as these numbers go up, you see that the response gets poorer and poorer, and here you have people who are not responding very much to these treatments at all. Right? So what do you do about this? You ask if you can perturb these conditions. Can you drive these cells that are in the Q1 state to cells like they would be in the Q3 state? And it turns out some of the new drugs can actually do that. There are these new drugs called direct acting antivirals. They directly attack some of the viral proteins and prevent replication. So they're not going through the immune system. They're targeting specifically viral proteins. And interestingly, it turns out at least the first generation of these direct acting antivirals didn't show much of a response. So this drug telaprevir is one of them. But when you use that in conjunction with interferon, the response rates went up enormously. So how does this work? And we have an explanation for that based on this dynamical systems view that we have, the double negative feedback loop. So what actually happens? If I have these direct acting antivirals, they block this replication process directly. But maybe it's not good enough for completely curing this cell of the infection, because this protection is somewhat weak. The blocking is not 100% effective. But if I now think about the interaction of this virus with the interferon system, then I realize if I have less of this, then I free up the block of the double-stranded RNA on the interferon system, and I therefore empower the interferon system to completely get rid of this. Right? So I move these bistable curves in a manner that the virus high ISG steady state now no longer becomes accessible. So now all of these states which were otherwise, all of these cells which were otherwise in this state have only this state to go to, and you get cure. Right? So this is how these drugs work, by manipulating these systems. So these antivirals alter these network properties and thus improve the interferon response. And today there are combinations of these direct acting antivirals which are very, very potent, and so within a matter of 12 weeks, if you put the individuals on these direct acting antiviral combinations, you can get more or less 95% cure rates of hepatitis, and therefore it has ceased to be a problem. So this is the first part of my talk. Let me then look at the other arms, the CD8 T cell responses. So the innate immune responses are good. They give you this quick and dirty way of fighting infections, but they don't always work, including in hepatitis C. I'll show you as we go along that you need other arms of the immune system to be active for us to see complete cure of this infection. So let's look at what the CD8 T cell responses are doing and how they are involved in the outcomes of viral infections. So people asked, like with the example of hepatitis C, why some people get cured of the infection and not others. So this is a problem that has been something that virologists have looked at for many decades. It's an old problem, best studied in this system of mice infections with this LCMV virus, lymphocytic choriomeningitis virus. And when you do this and follow what happens to this infection as a function of time, you find three classic outcomes. When the viral inoculum size is low, you get this virus level building up, but then you get clearance of the virus. So this is one outcome. If the virus levels are intermediate, the inoculum size is, then you see a lot of these mice dying because of what is called immunopathology. So if you look at the histology of the spleens of these mice, you see massive damage in these mice which had intermediate levels of this virus inoculum size. If you have high viral inoculum sizes, these mice don't die, but the infection isn't gotten rid of either. You get persistence, right? And so you see now the tissues are normal, but there's persistent infection. So the question now is, when do you see what? And this has been seen with many virus infections, these same three outcomes. So if one looks at, for example, these outcomes of viral infections in different settings, you see many different distributions, but of these very three outcomes. So with HIV infections, you see 100% of the time persistence. With measles, 100% of the time clearance. With hepatitis, as I said, 25% clearance and 75% persistence. Influenza, you get clearance or mortality, no persistence, and the level depends on the particular strain, whether it's H5N1 and so on. With hepatitis B, there's persistence in neonates and clearance in adults. With LCMV in mice, as I said, the outcome depends on the strain of the virus also. For example, if you have something called the Armstrong strain, then you get clearance. If you have clone 13, then you get those inoculum size dependent outcomes that I showed you in the previous slide. And SARS-CoV, everybody knows this, more than 95% people clear the infection. Small percentage they succumb to this infection. It turns out there's an even smaller percentage that now is showing persistent infections. So what determines these outcomes, and can they be controlled? So it turns out that as I said, virologists have looked at this for a long time. Many factors have been identified, right, over the years. For example, there's viral genetics. If you have, for example, this Armstrong clone versus the clone 13 clone, and they differ in their genome by only mutations in a few sites, you can get very different outcomes. Then there's viral inoculum size, as I showed you. And this, it turns out, is an important factor that determines whether clear the infection or not, and we'll look at this in a little more detail. Other cells of the immune system called NK cells, and there is the innate immune response, as I showed you in the hepatitis C case, and then there are host genetic factors and so on. And this list is a very long list, and it looks like, when you look at this, that the outcome is a rather complex, is a result of a rather complex interplay of a whole lot of factors. So it seems like it may not be easy for us to comprehend the factors that are responsible for the outcomes. And yet, it turns out there are just these three outcomes that you see. You either get clearance, you get immunopathology, or you get persistence. So if it's just these three outcomes, there must be something central that is deciding which of these outcomes is realized. And all of these other factors may therefore be altering whatever is happening with this central motif. So we asked whether we could understand what the central motif is. And when we looked at the literature, it turns out that it's the CD8 T cells that form this central component that is dictating these outcomes. And how do you come to that conclusion? Again, a quick survey of the literature, and it tells you this, that the CD8 T cells and their interactions with these infected cells or the antigen is what dictates most things. So for example, CD8 T cells, it turns out, are necessary for viral clearance. And the way you infer this is that you have mice which don't have CD8 T cells, and you then realize you can't just get rid of the infection no matter how strong the other arms of the immune system are. Persistent infections are associated with dysfunctional CD8 T cells. There's all this strong evidence in the literature. And what do these CD8 T cells do? How do they work? They get stimulated by antigen. So here's the interaction between the antigen and CD8 T cells that's beginning to manifest itself. And following this stimulation, they in turn kill cells that are infected by the antigen. So this is the business of clearing the antigen. And if you have sustained antigenic stimulation, this is another design in the immune system which I may not have the time to dwell upon, but it's an interesting design. If you have sustained antigenic stimulation, then the CD8 T cells become exhausted. That's the word that's used. And it turns out that this is an interesting design, because if you don't have this, then this can actually lead to massive tissue damage and pathology. Exhaustion is argued to be a design to prevent this immunopathology. And reversal of exhaustion, which can be done using drugs, can actually clear persistent infections and of course cancers. These are the immunotherapies that are currently being used and developed. So this set of observations tells us that it's the interactions of CD8 T cells and antigen that from these appear both necessary and sufficient to determine all of these outcomes. So how exactly is this going on? So our hypothesis based on this was that there is a dynamical motif that comprises the essential interactions of CD8 T cells and antigen and defines these outcomes. And all of the other factors that have been identified so far, they're important no doubt, but they influence the outcomes indirectly by modulating whatever happens in this dynamical motif. How do we test this? We build this dynamical motif and see what it tells us. This is the motif. It's a very simple motif. You have infected cells. Their numbers grow because they give out viruses. Those viruses infect other uninfected cells, and that's how this number increases. These then are seen by CD8 T cells. When that happens, they get activated. These activated CD8 T cells can kill these infected cells, and when you have a lot of these infected cells, then you can also trigger what is called exhaustion, which means that the power of these cells to kill these infected cells comes down. So now you can set up dynamical equations to describe what happens with this motif and what the outcomes would be. And it turns out you can show that again this admits bistability, and if you now plot the outcomes on a phase portrait, where I've shown here on the x-axis the population of infected cells and on the y-axis the population of these CD8 effector T cells, that you get these two stable steady states, one where your infected cells are very large, and that's the persistence state. The CD8 T cells are not very high in number, they are exhausted. And on the other hand, you have this state where your infected cells are very low and your CD8 T cells are potent. So this is the clearance state. And it turns out in this particular case, you also have this intermediate state, which is a saddle. It's an unstable state where both infected cells and these CD8 T cells are in high abundance. Now, this is telling us something about those experiments. If I think about what the initial inoculum sizes are, right? So I go from low inoculum size, high, intermediate inoculum size and high inoculum size. These trajectories here tell us how this infection would progress. So if I started here, you would have of course the virus level go up because the infected cells are going up, but then CD8 T cell responses are also mounted, and eventually you get rid of the infection. This is your clearance state. If you're here, similarly, you go to persistence. Right? It turns out it's this unstable state, the saddle, that underlies immunopathology. So that was the interesting discovery that we had. So if your initial conditions were very close to this separatrix, this is the boundary that separates the basins of attraction of these two stable states. Then you have these effector cells and infected cells, their activities poised in a manner that they both tend to rise. So the infected cells are not too large in number to cause exhaustion. At the same time, the CD8 T cells are not too potent for complete clearance to occur. So they both rise. The infected cells rise in number, the CD8 T cells also rise in number. And as you execute large trajectories along the separatrix, you reach stages where you have very high populations of infected cells and very high populations of these effector cells. And then you get massive tissue damage because all these infected cells are getting killed in the process. And that's your root of immunopathology. Right? And this tells you therefore that intermediate inoculum sizes will give you immunopathology. And you can see this playing out in these experiments. This is mice infected with LCMV. What is shown here on the left side is the population of mice that's persistently infected. On the right y-axis is the population that dies from pathology, and on the x-axis is the viral inoculum size. Low inoculum sizes, nobody is persistently infected, nobody dies, so everybody is cleared. Very high inoculum sizes, everybody, in this case again nobody dies, everybody is persistently infected. But somewhere in the middle you see death coming from immunopathology. And you can do the converse experiment where you keep the inoculum size the same, but you change the population of CD8 T cells, and you can do this by adoptively transferring these cells into these mice. And there now you are moving across the separatrix from the bottom to the top, right? And as your CD8 T cell numbers go from low to medium to high, you have the opposite effect. So if you have low CD8 T cells, everybody is persistently infected, nobody dies. High CD8 T cells, everybody clears the infection. And somewhere in the middle, in this particular experiment, you saw everybody die. Right? So this is now telling us how these infection outcomes come about. And this motif explains a large set of observations associated with these infection outcomes. Right? So this recapitulates these infection outcomes. So now just for a quick detour and also to make links with the previous example, you can ask what happens with innate immune responses. How do they really work, right? And it turns out, as I said, they modulate the outcomes of this very dynamical motif. So if I now think about innate immune responses, they are triggered by these infected cells and they curtail the replication of these viruses, and therefore this proliferation of these cells comes down. I can solve the same set of dynamical equations. Now you have a higher dimensional system, so I don't have a 2D phase portrait anymore. The phase portrait becomes a 3D system, but the essential features of the phase portrait remain. You have these two stable steady states, an intermediate saddle, and so on. Just for visualization, I can now project this onto the same 2D phase portrait that I showed you earlier. And what these innate immune responses do is they shift these lines. In particular, they shift the separatrix to lower values of the CD8 T cell responses. And this is understandable because now lower CD8 T cell responses are adequate for you to control the infection, because some of the controlling is now being done by these innate immune responses. So if I now think of an initial condition that was on the persistence side of this solid phase boundary, it would have gone here. But now if the innate immune responses are strong, which can happen when you introduce interferon, you move the separatrix to the other side, so that this point now is on this side of the of this phase portrait. You drive it to clearance. So this is how these innate immune responses seem to be working, modulating the outcomes that are otherwise already embedded in this particular dynamical motif. And you can see this playing out in hepatitis C virus infection. So here's an experiment with monkeys exposed to hepatitis C. Same inoculum size. Some monkeys show clearance, others show persistence. In these cases, you have the innate immune response coming up early, but you'll see even after the innate immune response comes, this is the place where the innate immune response comes up, where the uncontrolled replication of the virus is aborted. And then here is where CD8 T cell responses come up, the infection then dies out. Here you have a strong innate immune response, a weak innate immune response. This takes you to persistence, and this takes you to clearance. And the way you infer that is you look at the time at which you have this slope shift. That's the time at which the innate immune response is mounted. And then you look at the peak viral levels. If this is lower, then you have a stronger innate immune response. And if you look at all the macaques, and you can show also dynamically that this behavior can be recapitulated, you indeed see that this transition time is longer in the monkeys that showed persistence, and so also is the peak viral level. So what exactly is going on? If you have higher peak viral levels, when the CD8 T cell response is mounted, you are on one side of the separatrix, which takes you to persistence. Whereas if you have lower viral levels, then when the CD8 T cell response comes, your initial conditions are on the favorable side of the separatrix, so you drive it to clearance. So what your innate immune response is doing in this case is it's curtailing the viral load and therefore potentiating the CD8 T cell response to do what it's otherwise supposed to do. So this is the interplay between the innate and CD8 T cell responses. Now you can ask, what's the optimal load that the innate immune response must take, and then the CD8 T cell response must take, and so on. And those are questions that have not been answered. But you can see that there is a quick way of blocking the infection, which helps you limit the infection and also makes it possible for the CD8 T cell response subsequently to come and clear the infection. So that's my second part, and there are many applications to this. We can show that this same motif applies in the hepatitis C cure case, in HIV infections, and also in the case of SARS-CoV-2. I'm not going to dwell on it, but it also gives you some insight into engineering treatments. Right? So I can see that this helps me think about many interventions. I look at all the interventions that I have now. What I would like is for this interactions or the dynamical motif to be driven in a way that I empower the CD8 T cells, then I can get clearance. And how can I do that? I can do that by manipulating each of these entities here in this motif. So for example, if I lower the infected cell numbers, then I'm going to empower this. I can do that by drugs, I can do that by therapeutic antibody vaccines, and I'll talk a little bit about this in the coming slides. I can do this by increasing the strength of the CD8 T cells, and many T cell vaccines do that. I can do that by increasing the activation rate of these CD8 T cells, and this can be done using something called passive vaccination. I can do that by lowering exhaustion levels, and this is done using immunotherapies such as checkpoint inhibitors. I can increase the rate at which these CD8 T cells kill these infected cells, and things like CAR T cells, which is something that is very active in this particular campus, can do. Right? And what are all these things doing? They're actually trying to move the system from this side of the phase portrait to this side of the phase portrait. So you get a quantitative handle on how much you should intervene so that you make it then possible for the immune system to take over and get rid of the infection. Part 3. I don't know how much time I have. I have time, right? I look at the other arm of the adaptive immune response, antibodies, and I'll talk a little bit about how learning happens in the immune system. And I do this in the context of HIV infection. This is the time course of a typical HIV infection if it's untreated. You have the concentration of the virus in the blood of an infected individual as a function of time following the infection. The viral levels rise, they reach a peak. At this point the immune system has kicked in, it's controlling the infection, but it turns out in this case that you go to persistence. The infection is not gotten rid of. And this state lasts for a very long time. You'll see the change in scales here. You go from this initial fast dynamics in weeks to much slower dynamics over years. And then at some point, the immune system becomes so badly off that it succumbs to this virus continuously evolving and mutating, and you then have a surge in the virus in a condition that's called AIDS. The immune system is now so weakened that it makes the body susceptible to opportunistic infections, and the person dies. So to prevent this, of course, you treat the infection and it's done through antiretroviral treatments, and they work very well. As long as the individual is on treatment, the viral levels are very quickly controlled, they become undetectable and stay there for a very long time, as long as they are under treatment. If they don't miss the treatment, they comply and so on. But if you stop the treatment, then the virus comes right back and you have again progressive infection. Now, this is a serious problem. It implies that you need lifelong treatment. There have been no cases, and barring a few which I will discuss, where the infection has been completely cured. There's one very famous case of this person called the Berlin patient, an interesting medical case. It's an unfortunate individual who had both HIV infection and cancer. And for the treatment of that cancer, he needed a stem cell transplant. And the doctor, Dr. Hutter, he realized that he could transplant, he tried an experiment, he could transplant cells. And it turns out the cancer was of the cells that are targets of HIV. Right? You could transplant cells with a version of the cells that lacked a receptor that the virus required to get into those cells. So if you now do that, you would prevent subsequent infections of those cells by HIV. So they did that, and this person miraculously was cured both of the cancer and of HIV. And this has been repeated in a few other individuals, but very, very few, because you can't do stem cell transplants for HIV, especially when there's antiretroviral treatment. And besides, there are so many complications associated with that treatment that is not advised, right? So this is sort of the only case, if you will, and a few others after them, after this person, who have been completely cured of the infection. And by completely cured, what it means is that there has been no relapse. This person is now no more. But there was no relapse until the person passed away, many, many years after the transplant was done. So what's the alternative? The field has tried hard to come up with a cure, and they've realized that maybe that's not the best way to go about doing things. Can we come up with alternative strategies which will give us what is called functional cure? So you don't worry about completely getting rid of the virus. Can I come up with an intervention which I can use for a short period of time? Then perhaps by the ways that we discussed in the previous set of slides, stimulate the immune system so that control of the virus can be brought about and progress of the infection to AIDS can be prevented. So can this be done? This is the question that people have been asking. And one of the most promising therapies that has been tried today is this thing called antibody therapy. And this is something that I think most people are familiar with from the COVID times. You've got this plasma therapy. You take somebody else's blood and then inject, because that has antibodies that are supposed to block viral infections. So that's what you do here. I mean, that's not how you do it. That's just a cartoon. You put in antibodies, right? And what the antibodies do is they block viral infections. So here's your virus, these are these antibodies, they coat the virus surface, and then they don't allow the virus to get into these cells which it is supposed to enter and replicate. So the viral levels come down, and you hope therefore that the infection is gotten rid of. This is a treatment under investigation. There's some very interesting observations that came about when people tried this for the first time, and I'll show you that, and that will give us lead into this business of immune learning and memory. This is an experiment that was done in 2016 where this particular antibody was injected in individuals at different doses. So the different individuals were given these different doses, and this is the concentration of this antibody as a function of time, right? You add this at time 0, and by 16-20 weeks this antibody is flushed out. So that's the experiment. You give one dose and you watch. And what you ask is how potent the antibodies are before you administer this and how potent the antibodies are after this is completely washed out, right? So what you're looking at is the strength of the endogenous antibodies made by the body themselves before this treatment and way after this particular treatment. And here's the interesting observation. This, what is shown here, each bar is an individual who was subjected to these treatments. These yellow bars are controls, so they were not given this antibody, they were given placebo. These blue bars are people who are given this particular treatment. What is shown here is the difference in the potency of these antibodies to block viral infections relative to the potency of these antibodies. Right? So in the untreated controls, there's no difference. There's no statistically significant difference. Some are up, some are down, and so on. But if you look at the individuals who are given this antibody, you find that there is a significant increase in the endogenous antibodies' ability to neutralize virus relative to what they had before this treatment. And this increase, interestingly, is much higher in the individuals who are not on antiretroviral treatment. Right? If you were on antiretroviral treatment and given this, then the gain was there, but much smaller than this. So how does this happen? Right? So these antibodies are supposed to block the virus, get rid of it. How are they altering the endogenous antibodies' efficacies? This is the question. So for this, we must look at how these antibodies are produced and what processes might be affected by these passive administrations of these antibodies. Antibody production happens in this region called the germinal center reaction. These are temporarily assembled structures in our lymph nodes in response to infections. Here are B cells, which are the cells that eventually produce these antibodies. They get selected, they get evolutionarily selected and trained to produce high affinity antibodies. This is Darwinian selection on fast track, right? So you will see how this works. These germinal centers are divided into a light zone and a dark zone, and these names come from the density of cells that they have in these two regions. You have here something called follicular dendritic cells. These cells have on their surfaces receptors that present to other cells these antigens, in this case HIV, attached to an antibody. So the surface has a receptor, to that is attached an antibody, and to that is attached its antigen, which is the virus. You have here B cells, right? These are the cells that eventually will produce these antibodies, and these B cells have, each B cell has a different B cell receptor, and this is one of the most sort of interesting parts of our immune system, because these B cell receptors are produced by massive shuffling of the genes that are responsible for the production of these receptors. So massive variation comes about. So each B cell has a different B cell receptor. Now these B cells, these B cell receptors are actually transmembrane versions of the antibodies. So they are exactly like the antibodies. They can latch on to this antigen and now they pull. And if they are successfully able to acquire the antigen, then they get stimulated. And when that's, so just to give you an idea, the efficacy of this pulling is related to the binding affinity of the B cells with respect to their antigens. So if I describe these antigens as bit strings, let's say, just for convenience sake, and the surfaces of the B cell receptors as complementary bit strings, then the number of filled bit strings gives you an idea of the affinity. This is low affinity B cell receptor, high affinity B cell receptor, and so on. This acquires the antigen, this doesn't, and therefore this gets a signal that stimulates it. This dies. Now, when many of these cells acquire these antigens, they compete for a second signal. In this particular case, a signal that comes from what is called a T follicular helper cell. This is an interesting process also. Many of these B cells which are stimulated, they simultaneously attach to this B cell that also presents antigen, and it now gives a signal to that cell that has acquired the most antigen to proliferate. So you see now there is local competition that is getting resolved through this particular signaling event. These cells now that receive the second survival signal, the others that don't die, they suffer one of three fates. They can either get out of the germinal center, mature into what is called a plasma cell, and keep producing large numbers of these antibodies, which are now secreted versions of the same B cell receptor. Otherwise, they can go to the dark zone, where they now proliferate and mutate, and when they mutate, they now cause variations in their B cell receptors. So new sets of cells come up. So now you can see what you had was this kind of a B cell receptor. Because of mutation, you have produced something that has even higher affinities, right? You can also get memory cells, which I think I forgot to draw here. So now these set of cells which are mutated and proliferated come back to the light zone, and they are subjected now again to the same selection process, right? And so now you can, in this case, these cells are competing. This has a higher affinity than this cell, so it preferentially acquires the antigen, gets stimulated, and again suffers one of these states. If it becomes a plasma cell, it starts now producing antibodies that have even higher affinities for these antigens than the ones before. This is the learning process. This is how you improve the affinity of the antibodies that are getting produced. It's beautifully genetically encoded and has underpinnings in all of these processes. It turns out that there were models that were constructed that showed that if you have to have this affinity be optimally manifested, then this separation of these light and dark zones were essential. Here is where selection would happen, here is where proliferation and mutation would happen, right? And this was predicted first by a theory, and then when people went and looked at these germinal centers, they found that this separation of spatial separation was there, and the cyclic re-entry, as they called it then, was observed. Anyway, so this continues. This process of increasing affinities is called affinity maturation. Now, it turns out that these antibodies that are produced, they can also feed back into the germinal center. So this is the complexity, right? When they feed back into the germinal center, they also compete for antigen with the antibodies that are used for presentation. If these have higher affinities, they will then replace these antibodies with themselves and make it now much harder for the B cells to acquire antigen, because they are now holding the antigen much more tightly, and therefore you increase the selection stringency because of this feedback process. This is what we believe these passively administered antibodies are doing. So if I now use antibodies like the 3BNC117, comes into the germinal center, there it replaces the presenting antibodies with itself, makes selection much harder, and therefore now selects for B cells that can produce these higher affinity antibodies. And now, even after these administered antibodies are gone, you are left with these antibodies that are produced by endogenous B cells, which have higher affinities. They continue the selection process, taking off from the place where it was left off by these antibodies. So you have permanently altered the selection landscape inside these germinal centers. Does this really work? So we said, let's look at some simulation. We did some simulations. Our hypothesis was this, that you have these passive antibodies that increase the selection stringency. Just to make this clearer, these are the receptors on the follicular dendritic cells that present these antibody antigen complexes, and passively administered antibodies, if they have higher affinities, they can preferentially take this and become themselves these presenting antigen antibodies. Now if I have an FDC that is interacting with a B cell, this B cell is supposed to acquire this antigen. It's easier for it to acquire with the low affinity antibody presenting it, but if you now have this presenting it, it makes it much harder. So this is the selection stringency that we are talking about. So we can now set up stochastic simulations of this germinal center reaction using what are called in evolutionary theory Wright-Fisher simulations. So these are discrete generation simulations that tell us how the affinities of these antibodies are evolving, where each of these cells is considered an agent and is subjected stochastically to different fates based on all the descriptions that I showed you. I won't go into too many details, but I'll tell you that these extraction probabilities can be computed through affinity calculations, and they become proportional to the differences in the affinities that these individual antibodies have to the antigen. And then you can model also these T cell help processes as the probabilities that become proportional to the antigens that a cell has acquired relative to the mean levels that are acquired by all of the cells in that generation. So when you do this, you can model all of these processes and look at what the outcomes are. And when you do this, you find that in the germinal center, interestingly, there is this quality quantity trade-off. And it's also evident from all the things that I just said. What is this quality quantity trade-off? If I use antibodies of very high affinity passively, right, then I make the selection stringency very high. Then I have cells that have even higher affinity for the antigen than the administered antibodies getting selected, but there are far fewer of those cells than if I had the selection stringency weaker. So I produce antibodies of high quality, but there will be far fewer of them. Right? So this is the quality quantity trade-off, and you can see that playing out. If I use passively administered antibodies of low, intermediate or high affinity, then the germinal center output, right, goes down, but the quality of the output improves. right? And this you can see, this is the prediction of the simulation, this you can see also captured almost perfectly in mice experiments when this was done, right? So you can see this quality quantity trade-off playing. The other angle to all of this is that you can tweak this quality quantity trade-off by altering the amount of antigen that is present in the germinal centers. If I have a lot of it, and even if they are high affinity, then I have each B cell getting many more chances to acquire antigen. Then again I can have lower affinity B cells getting selected even if the antibody is administered or of a much higher affinity. And this you can see from other kinds of experiments where antigen now was administered at different levels or different dosing protocols. So you have an exponentially increasing antigen level. So this also presumably also changes the level of antigen available in germinal centers in the same proportion. That's the expectation. Exponentially decreasing, constant, or you give everything in one shot, making sure that the total amount of antigen that is administered is constant. And you see from these experiments that this profile where you have an exponentially increasing antigen level elicits the best antibody response. And this happens because as your B cells are evolving, they're getting better and better in their affinities, but then you're also now making more and more antigen available so that you're allowing these slightly lesser affinity B cells also to continue to get selected, right? And so you improve quality, you improve quantity, and that's what this shows. And you can recapitulate this also from our simulations. So you now have these two parameters, the affinity of the antibodies that you're using and the level of the antigen that is present, which you can now use to control these outcomes. And you can see that immediately this explains what happens here. You can understand that because of this antibody administration, the affinity of the antibodies that are produced later is going to be higher than what you had before because you have now altered the selection stringency. But you can see now this business of individuals off versus on ART having these different effects. Right? In this case, because there is a lot of antigen available, there's high amounts of virus in the body, you can see selection stringency, although it's important, is less penalizing in terms of the quality quantity trade-off, and therefore you see better neutralization responses. Whereas here you see poorer ones, so high antigen and low antigen. This you can now use to optimize the way in which you administer these antibodies, passive antibodies. So you can use these simulations to ask, for example, this kind of a question where I have 3 doses that I'm going to administer, and I have at my disposal all these different types of antibodies, and this is a realistic scenario. These things exist, and these 3 doses are also what many times are used. What's the combination that I should use to maximize my antibody response? And why is this useful? If I get a good response, then I can be off therapy and hope that these antibodies exert long-term control. And there are these individuals who are called elite neutralizers, right, who have these potent antibodies spontaneously produced, and they can control the virus without treatments for a very long time. They're a very small fraction of the individuals though. So the question is, can we reproduce that phenotype through interventions like this? Yeah, I'll stop here. So you can run these simulations and you can see now what the overall antibody output would be as a function of the availability of this antigen, which is the level of the virus in the system. And what you find is that the optimal dosing protocol is not the same across all individuals. So this is now the various protocols that I mentioned, all low affinity, all high affinity, and some combination of low, intermediate and high affinities. And depending on what the level of the virus is, which is a measure of your antigen availability, you find different combinations being optimal. So this is telling you that these passive immunizations should not be used as a blanket one size fits all formula. They have to be tailored to suit the antigen availability that is there in these individuals. Right? So this is where you have this learning that is going on, and you can use your understanding of that learning process to engineer it through interventions which can then potentially allow for long-term control of the virus to be established. And there's lots of promise that people are seeing through both mice experiments and human clinical trials for the use of these passively administered antibodies in eliciting long-term remission of HIV infection. So at this other section, I'm going to skip in the interest of time. But here, what I was going to tell you is that there is this business of the immune repertoire. So this is a great question to ask. How capable our immune system is or to fight everything that we might see. So do we have power in us to fight every pathogen that we might encounter? Right? Or are we by design vulnerable to some? This is a question that, it's an interesting question that people asked, and this brings us to what is called as the immune repertoire. Right? It tells you how broad our immune response is, how diverse a set of pathogens it can handle spontaneously and effect a cure, and that gives you an idea of how to use vaccination, because that's the memory that you can create based on the repertoire that you have to deal with upcoming infections. And just to keep, in the interest of time, I'll tell you what we did with that business in trying to predict vaccine efficacies in these COVID-19 scenarios. You saw all these numbers, right? Where we had this Oxford vaccine which had 70-80% efficacy, and Pfizer's had 95% efficacy, and so on. And you can use these ideas of the immune repertoire, and in this particular case, this business of an immune landscape in which antibodies are produced to explain these outcomes, these efficacies of the vaccines quantitatively. So I'll skip this story in the interest of time, but go to the very end to tell you that you can again think about these design principles that tell you what these landscapes are of the antibody responses that are produced, drawing again from these Germinal center reactions that we discussed. And you can show from simple modeling ideas that you can actually recapitulate the entire set of vaccine efficacies that have been seen with all of the approved vaccines for COVID. Right? So this gave us also a handle on how to engineer these vaccines and how to also think about markers of protection. In this particular case, this was a blood-based marker which could tell you how to predict efficacies that were otherwise seen in phase 3 trials. So you can now use something, you administer these vaccines to a few individuals, draw blood, look at the antibody responses in them, and you can instantly predict what the efficacy would be of that particular vaccine if it was run in a phase 3 clinical trial. So you can obviate the need for phase 3 clinical trials with this understanding of how vaccine efficacies come about. So I'm going to stop in the interest of time and leave you with this outlook, which says that there are these intricate engineering designs that govern the immune system. Some are known, some are yet to be unraveled, many of them remain unknown. What our own and many other colleagues in the field have, the work of ours and other colleagues has done is help us think about some of these parts, right, in isolation even. The question of course is how to piece all of these together and see if one can construct a holistic view of the immune system that then allows us to engineer the responses that are administered or that are elicited to pathogens that today remain incurable. So thank all of the colleagues, students and collaborators who helped us do some of this work, sources of funding, and once again thank you all very much for this wonderful invitation, for the lovely time that I've had the last 2 days, and for reliving the memory of Professor C.V. Seshadri. Thank you very much. Thank you.
Antibody response that you were talking about and how the affinity changes.
Yes.
So, if our immune system has the capacity to increase the affinity, even the viral would be changing its antigen. So, how do we account for that in the system?
That's a great question. So there is always this co-evolutionary sort of arms race that's going on between the virus and the immune system. In many cases, people try to engineer vaccines that will elicit antibodies to what are otherwise called conserved regions on the viral surfaces. So most of the HIV antibodies that are used for these treatments, they target conserved regions on the virus. These are regions where if the virus mutates, it will have serious fitness penalties and therefore it's not sustainable. But otherwise, if you don't do that, and if you simply think about affinity maturation as a consequence of this evolutionary process, then there's a moving target. The virus is evolving, and the antibodies are also evolving, and simultaneously you see clones of B cells that target the dominant strains of the virus coming up. When the virus evolves, those strains decay, new strains come up, and so on.
First is when you talked about this, thank you. So the first is where you talked about the saddle point distribution of that persistence and clearance. So is there any study that has considered the role of T regulatory cells, because that also comes into the picture when the inflammation goes up? So is there any study that has considered?
There are several studies that consider regulatory T cells,
Okay.
and they, for us, also do the same job in some ways of what exhaustion does. Because if you have a lot of the cells unable to control the infection, or if the immune system has sort of already mounted a significant response and not a whole lot is going on, then there are all these regulatory mechanisms that kick in that prevent serious immunopathology and other tissue damage. So these regulatory mechanisms are factored in, and they sometimes help when you actually want the immune response to taper off, but they can be counterproductive if you want the immune response to be aggressive, but then these regulatory T cells come in and try to suppress it. So there are also methods that people are constructing that can engineer the timings of these responses correctly.
Okay. And the next question is, when you talked about the affinity maturation, you said the antibodies can come and again bind to the antigen in that region. So is that a potential, can that be a potential cause of autoimmunity? Can that be considered as autoimmunity, because the self antigen is targeted against the antibody? So can you consider that?
It's not self antigen that I talked about, it's the antigen that comes from the pathogen. It's against that antigen that the evolution is taking place.
Okay.
Autoimmune responses do happen, it's not like they don't, but in this particular case that was not the case.
Okay, thank you so much.
Maybe a couple of questions, and in case of time, we have tea outside right after this, so we can take some of the questions outside. But 2 more questions and then
Hello, sir. Nice talk. As you said that the antigen concentration in the, let's say the mice, is very important to generate different levels of antibody production when we exogenously supplement the antibodies. Can we control the antigen in the body by using the antiviral treatments?
Great
Or will it help?
No, that's a great question, and people worry about this a lot. So if you use very strong antiviral drugs before you administer antibodies. So today, especially in the case of HIV, the antibodies are being used in conjunction with antiretroviral treatments, because you can't otherwise completely get rid of it, except if you do it in monkeys and so on. So when you do them in conjunction, if you use antiretroviral drugs that potently suppress the antibodies, suppress the antigen, then the passively administered antibodies don't have as much of an effect. So people talk of these therapeutic windows where you give the drugs to bring the levels down, but not too much that the antibodies fail to work.
So as you were supplying the exogenously antibodies in a increasing fashion, we can also use the ARTs in a decreasing fashion.
Perhaps. Perhaps.
Thank you so much, sir.
Hi, sir.
I can't see.
Yeah, you're here.
Oh, there, yeah.
Yeah. A very interesting talk. I just had a question about the saddle point again. So in the saddle point, 2 factors were considered: CD8 T cell and the viral load. But since there is a deviation, is there any intelligence or crosstalk between the viruses and the host defense that could actually drive away the model elsewhere?
Try to do what, sorry?
Some crosstalk between the host and the virus responses, and that could actually drive away the model elsewhere. Exceptional cases.
Drive the model elsewhere.
Yeah.
That's a caricature. The model obviously eliminates a whole lot of other processes that are in place, which we have not considered, right? Because it's a minimal model which takes into account what we call the essential interactions. But there are obviously possibilities where other interactions which have been ignored in the model become dominant in certain scenarios. Then many of these things will not work. For example, even innate immune responses. I showed you some case where you can think about the innate immune responses as modulating these interactions. But there are a whole lot of other things. NK cells, you can have, people have shown that you can have converse effects, counterintuitive effects, where you increase the NK cell numbers and you get worse responses and things like that. So there are these scenarios. There are extreme scenarios, like you said, and which perhaps these models at the level of complexity that they are dealing with cannot handle.
So is there a way somehow we can add on that to the model
yes,
later?
you can. But it makes the model more complex, and then the intuition that you expect becomes harder to come up with.
Okay, yeah, thank you.
Okay, thank you for the privilege. So very nice. Very simple question. I was just looking at the model prediction angle. So what are the mathematical framework you use for going from blood marker to virus efficacy? I mean, what kind?
Blood marker too?
The the vaccine efficacy rather, sorry. In the last COVID-19 case or in the previous?
Yes, so the blood marker in the vaccine case, the COVID-19 case, is the neutralizing efficacy of the antibodies that come from the blood of a vaccinated individual.
Right.
So I can draw blood and perform an in vitro assay where I have cells that are susceptible to the virus.
Okay.
I expose them to the virus in the presence of this antibody and look at how much of the infection is blocked by these antibodies. That's something that you can do.
Oh, okay. So it's as simple, but that is in vitro. Does it work in the real life?
Yeah, that's the correlation. So that's the efficacy that now predicts perfectly the phase 3.
That's what in vivo also it replicates.
That phase 3 is now actual population level trial.
Right. No, so there are variations which goes from, I mean, when you do in vivo clinical trials, no, so those the model is able to supersede that, and
yes,
so what is there in the model that helps to be so robust across individuals? And
great question. I didn't have the time, unfortunately.
Yeah, that's why I thought you will take a minute to recover the lost time.
Thank you. So there is variability that comes up from the kinds of antibody responses that are elicited to a particular antigen. It turns out there is a collection of antibodies that can all neutralize a particular antigen, and different individuals sample different antibodies from that large pool. And that's the variability that describes differences in the antibody responses elicited by different individuals to the same antigen. And we actually constructed that landscape, the entire pool of antibodies that can be elicited in response to the SARS-CoV-2, for example, in this case, spike antigen. And then by drawing random samples from that as representatives of the responses that different individuals elicit, you can predict correctly the phase 3 types of responses that people see in clinical trials. Thank you.
Great. Let's thank Narendra once more time for this wonderful talk.
Thank you very much. Thank you. Thank you.
Before we end, let me request Mahesh again to do the honors and give Narendra's memento. All right. Just before we leave, let me just before we head out, let me just thank all the people involved who made this possible. Starting off with again Narendra, you thanks for accepting our invite and spending 2 days with us and the talk today and yesterday. This was great fun. The Institute Distinguished series has been made possible by efforts of Professor S.K. Gupta, who's now retired. Thanks to him. Thanks to all the donors of the C.V. Seshadri Endowment Fund for their contributions. Like to thank everyone involved, including people from the State Office, VMCC staff, CD8 team, people from the NACR's office, particularly Sneha and Manjula for their help and for all the help with the logistics. Thanks to all the staff in the Chemie department for putting up of all the arrangements for this talk, especially as always Sudhirji, and thanks all of you for showing up, and we have tea outside, so please join. And if you are, I know we cut you short with the questions, if you have more, we have a few more minutes, he has to leave soon, but please join us for tea outside and some snacks. Thanks again.